I2BC is a research center where more than 500 scientists aim to decipher the organization and the functioning of living cells at different resolution, at different time scale, and in different conditions, for example during infections, pathologies, or different stresses. We use a set of complementary approaches and cutting-edge technologies to perform our studies. Our objectives are to address important questions on four major themes: the architecture and dynamics of cellular structures and their dysfunctions, the processing of genetic information and its dysfunction, the interaction between host and microbes, and the bio-inspired engineering for health, energy, and the environment. To reach these objectives, we want to implement every year 4 ambitious research programs by federating expertises from research groups and technological platforms, providing seeding resources from I2BC to each selected program, and securing mid-term funding of the project and developing partnerships. In 2021, the 4 programs concerned: the crosstalk between 2 major cytoskeletons found in eukaryotic cells, the effect of RNA modifications called epitranscriptomic marks on the cellular fate of RNA molecules, the compartmentalization of living cells in the absence of membranes, and a new engineering method to assemble macromolecular complexes with different applications.
Our institute is a deep phenotyping facility. Indeed, we push MRI technology to its limit to get more insight about physiopathology, aiming at the stratification of the patient populations into homogeneous groups with a simpler disease causality.
Our challenge is to develop a technological pipeline to predict and engineer the structures of macromolecular complexes. Those macromolecules can be natural proteins with a function in the cell that we want to optimize or inhibit. They can also be artificial proteins, so-called biologics, specialized in mediating protein-protein interactions, such as nanobodies. We are developing structural bioinformatic tools and methods to model and engineer the structures of those complexes. We have recently explored the combination of deep learning approaches with high-throughput metagenesis technologies to improve the accuracy and reliability of our predictions. This project, for instance, was supported by the Sanofi iTech Award. As an application of our design strategies, we have successfully developed protein-protein interaction inhibitors starting from a natural peptide that we managed to design with a nanomolar high affinity for its target. From there, we were able to further engineer this compound to turn it into a non-natural peptidomime, retaining the same affinity but with a much higher stability in human serum and lower immunological responses.
Hi everybody, I'm Michel Mazella. I'm working at the French Nuclear Agency and I am an expert in microscopic simulation and high-performance computing. In particular, we are developing original tools to investigate matter at the microscopic level and to understand the microscopic factor modulating the strength of the force at such level of the matter. We are already collaborating with groups like L'Oréal, personal care and cosmetics, Sanofi, industrial pharmaceutical groups, and a power supplier like EDF in France. Obviously, we will be very pleased to collaborate with you if you are interested by such tools. Thank you.